Good morning. It's October 8th, and this is your daily brief in science. Here's everything you need to know. On October 7, 2024, the Nobel Prize in Physiology or Medicine was awarded to U.S. researchers Victor Ambros and Gary Ruvkun for their groundbreaking discovery of microRNA and its crucial role in gene regulation. This prestigious award includes a diploma, a gold medal, and a shared $1 million prize, recognizing their significant contributions to the field of science. This announcement marks the beginning of the annual Nobel Prize season, which will also include awards in physics, chemistry, literature, and peace throughout the week. Researchers Anne Houdou-Sejoui and Antonin Morillon from CNRS/Curie have highlighted the importance of Ambros and Ruvkun's discoveries, emphasizing their profound impact on the field of gene regulation. In a related development, John Hopfield and Geoffrey Hinton were awarded the Nobel Prize in Physics for their foundational work in machine learning using artificial neural networks. Hinton's invention of a method for autonomously identifying properties in data has facilitated advancements in tasks such as image recognition, proving essential in the current landscape of artificial intelligence technology. While Hinton's contributions are celebrated, they also bring attention to his controversial views on the risks associated with AI. Hinton believes that AI will greatly influence civilization, enhancing productivity and healthcare, and likens its potential impact to that of the Industrial Revolution. The Nobel Committee acknowledged the dual nature of machine learning, emphasizing both its benefits and the ethical concerns it raises regarding its rapid development. As the Nobel Prize announcements continue, predicting the recipient of the Peace Prize remains particularly challenging amid numerous global crises. Current issues include the war in Sudan, famine risks, and rising military spending, all of which complicate the landscape for potential Peace Prize candidates. In an intriguing revelation from the world of marine biology, a study published in Current Biology has uncovered a remarkable ability in certain comb jellies. Researchers led by K. Jokera from the University of Exeter and the National Institutes of Natural Sciences found that Menemiopsis leidii, commonly known as sea walnuts, can actually fuse their bodies to create a single organism. This discovery originated during routine research at the Marine Biological Laboratory in Woods Hole, Massachusetts, when scientists noticed a larger-than-normal sea walnut. This prompted further investigation into the phenomenon of fusion among these creatures. Initial observations of the fused jellies indicated they had 2 back ends and 2 apical organs, leading researchers to hypothesize that fusion had occurred. To test this hypothesis, Dr. Oscar Arenas and his team replicated the conditions by injuring pairs of sea walnuts and pinning them together overnight. Remarkably, they achieved fusion in 9 out of 20 pairs. The fused jellies demonstrated rapid neural integration, responding collectively to stimuli within 2 hours, suggesting their nervous systems had synchronized. A particularly fascinating finding was that when one jelly was fed fluorescent food, the particles subsequently appeared in the digestive system of the other jelly. This indicates a lack of tissue recognition, allowing them to share resources. Understanding this fusion process could have significant implications for regenerative medicine, shedding light on immune system mechanisms and the potential for tissue grafting. The absence of allorecognition in these sea walnuts raises important questions about their evolutionary biology, necessitating further research into their neurology and genetics. Future studies will explore whether other species of comb jellies, such as Balanopsis mikado, possess similar fusion capabilities. While this fusion behavior is fascinating, it is believed to be extremely rare in the wild, as it requires specific conditions, such as close proximity between injured individuals. The findings of this study not only highlight the unique biological traits of comb jellies, but also position them as valuable models for understanding evolutionary processes and improving human transplant techniques. A recent study published in Nature Biotechnology has unveiled new techniques in genomic profiling and cancer diagnostics that could transform the landscape of precision oncology. This research, led by authors including Song, J. et al., emphasizes the importance of comprehensive genomic profiling, or CGP, in advancing personalized medicine for cancer treatment. At the heart of the study is the OncoIndx platform, a next-generation sequencing assay that identifies critical mutations to guide therapeutic decisions. OncoIndx has achieved an impressive clinical sensitivity of 98% and a positive predictive value of 100% for actionable genomic variants, indicating its potential effectiveness in clinical settings. The study also addresses the enhancement of liquid biopsy techniques through the high sensitivity and specificity of the OncoIndx assay, which could help overcome significant implementation challenges. Additionally, the research explores pooled CRISPR screens that allow for the simultaneous measurement of cellular responses to genetic changes using DNA-encoded barcodes. A novel technique called CRISPR map has been introduced, improving the detection of cellular barcodes through optical readout. This advancement aids in analyzing various cell types, including primary and stem cells, and has shown the capability to accurately replicate known phenotypes associated with specific base edits. Furthermore, single-cell multimodal profiling, which assesses both proteins and RNA, is highlighted as crucial for a deeper understanding of cellular functions. The OncoIndx assay targets major exons and selected introns of 1,080 cancer-associated genes, analyzing complex biomarkers such as single nucleotide variants, copy number alterations, and specific gene fusions. The assay also provides insights into tumor mutational burden, homologous recombination deficiency, and genomic loss of heterozygosity, which enhance its clinical relevance. Additionally, a new CRISPR-based assay developed in the study demonstrates subatomolar sensitivity for recognizing and amplifying target mRNA, showcasing its potential for future clinical applications. NASA's OSIRIS-REx mission has made a groundbreaking contribution to our understanding of the solar system. On September 24, 2023, the spacecraft successfully returned a sample of dust and rocks from the near-Earth asteroid Bennu, delivering 4.3 ounces, or 122 grams, of material back to Earth. This achievement surpassed the mission's initial goal of collecting 2 ounces, providing researchers with valuable insights into the early solar system and the potential origins of life on our planet. Initial analyses of the asteroid material have revealed high quantities of carbon-based compounds and hydrated minerals. These findings suggest that asteroids like Bennu may have played a significant role in delivering essential components for life to early Earth. Additionally, researchers identified magnesium-sodium phosphate in the sample, indicating that Bennu's origins could be more complex than previously thought, possibly originating from a larger, water-rich celestial body. The majority of the asteroid sample is curated at NASA facilities, while portions have been distributed to key research institutions and select museums to foster public engagement with this historic mission. Following the successful return of the Bennu sample, the OSIRIS-REx mission has transitioned into a new phase called OSIRIS-APEX. This new mission aims to study the near-Earth asteroid Apophis, which is set to approach Earth in 2029. Led by Dani Mendoza de la Giustina, the OSIRIS-APEX mission will gather data on the interactions between near-Earth objects and Earth's gravitational field, a crucial aspect for developing planetary defense strategies. Furthermore, the mission's data may provide insights into a potential fifth fundamental force of the universe, as suggested by a study from an international team led by Los Alamos National Laboratory. Co-author Sunny Vagnozzi emphasized that their findings offer some of the tightest limits on potential fifth forces, which could aid in the search for ultralight bosons and dark matter. This area remains poorly understood, as dark matter constitutes about 85% of the universe's total matter, but its particle composition and forces are still largely unknown. Lead author Yu-Dai Tsai noted that tracking data from Bennu could enhance our understanding of the standard model of physics, gravity, and dark matter. The research team utilized ground-based tracking data collected before and during the OSIRIS-REx mission to investigate extensions of the standard model, further advancing our comprehension of the universe. A recent study published in JAMA Network Open has revealed a significant breakthrough in predicting treatment responses for metastatic prostate cancer. The research highlights the potential of measuring circulating tumor cells, or CTCs, at the onset of the disease to forecast patient survival and treatment efficacy. CTCs are rare cancer cells that are released from tumors into the bloodstream. While they have primarily been studied for their predictive value in advanced stages of cancer, this new research marks a pivotal moment in understanding their role at the initial diagnosis of metastatic prostate cancer. Conducted as part of a Phase 3 clinical trial supported by the National Cancer Institute, the study involves collaboration from multiple institutions across the United States. The study utilized cell search technology to analyze blood samples from 503 men participating in a new drug trial. The findings indicated a clear correlation between higher CTC counts and shorter median survival rates. Specifically, men with 5 or more CTCs had a median survival of 27.9 months, while those with 1 to 4 CTCs had a median survival of 56.2 months, and those with no CTCs had over 78 months. Lead author Dr. Amir Goldkorn emphasized that this is the first study to assess CTC counts at the initial diagnosis of metastatic prostate cancer, providing essential insights into patient outcomes. The research aims to enhance clinical trials by identifying patients who may benefit from more aggressive treatment strategies based on their CTC levels. Additionally, Goldkorn's team is working on a new blood test to analyze the molecular composition of CTCs and tumor DNA to further improve treatment matching. Currently, once prostate cancer metastasizes, treatment options shift from surgery or radiation to systemic therapies focused on prolonging survival. The overall goal of this research is to enrich clinical trials by pinpointing patients who would benefit from more intensive treatment approaches, ultimately aiming to improve survival chances. This has been your daily brief in science. To read more about these stories, follow the links in the episode bio. You can also subscribe to these updates via email at www.brief.news. For more daily podcasts about the topics you love, visit brief.news/podcasts. Tune in tomorrow. We'll be back with everything you need to know.